Today's presentation is De-risk your program with evidence-based secondary pharmacology insights. Our speaker is Dr. Will Redfern. Will is currently a Vice President of Quantitative Systems Toxicology and Safety at Certara. At Certara, he leads the OSTS team to apply computational approaches to assess the safety of pharmaceuticals and other chemicals. Will is an experienced safety pharmacologist, having worked at Syntex, Quintiles, and AstraZeneca. He's also the former president of the Safety Pharmacology Society. Will, welcome to today's event. I'll now turn it over to him to begin the presentation.
Thanks, Suzanne, and good afternoon and good morning, everybody. So if I can change to the next slide, we're in business. The mouse on the right screen. There we go. Okay, so hopefully let's change the next slide. So this is quickly to say who Certara is and what we do. And probably the key message from the text at the top of this slide is that our software has been involved in over 90% of all new drug approvals by the FDA since 2014. And some of you will recognize our software products, including the PK/PD platform Phoenix, which is widely used, the SimSip PBPK simulator. We also have a SIVA in vitro analysis toolkit, cardiac safety simulator, and also D360, which is a database and informatics tool. And QSTS tools are in development. So to save some bandwidth, I'm going to stop sharing the webcam and move on to the next slide. Oops. Just managed to lose the presentation there. Okay, right. So just to focus on our team, which is called Quantitative Systems Toxicology and Safety, or QSTS. And we're a team of about 12 scientists, so we could call ourselves safety data science scientists. And it's really a good mix of modelers and safety scientists. So we're involved, first of all, we've got a consortium going with Amgen at the moment, and you're welcome to join this consortium. This consortium's also involved in developing products for ToxStudio, which I'll come on to, and we're going to talk about secondary intelligence today. The team is also involved in several public-private partnerships, all of which are EU-funded initiatives. So we're working with several pharma companies already via these consortia. And then we also offer technology-enabled consultancy. And this is where we use the products either generated within QSTS or our colleagues elsewhere in Certara UK. So to talk you through the ToxStudio concept, so this is really a core interface which integrates knowledge and modeling. So it will take data from in vitro readouts, will also interface with modeling modules, including ones you may have in-house. We'll take on board other data. We'll talk to SimSip, the big PBPK engine, and provide visualization and data output. Now we can see already that we can get input via the SIVA toolkit and also SimSip Animal. These software packages are already available. And then the new kid on the block, which I'm going to talk about today, is secondary intelligence software. So this is the problem we're trying to address, essentially. We're talking about safety-related attrition. And as you know, safety issues, preclinical and clinical, continue to contribute about 25% to the overall attrition of drug projects. And this is data that I've collected from sources dating back to the 1960s until relatively recently. And you can see there's no upward or downward trend. It's pretty stubborn around 25%. Now, this is being addressed by large pharma in 4 ways. Anticipation of target-related toxicity by target safety profiling. That's before you've even synthesized any compounds. Early de-risking, so that's avoiding molecular structures and physicochemical properties associated with known toxicities. And then hazard detection from in vitro screens and assays, from the use of omics and biomarkers in vivo. And then risk assessment, where you have a synthesis of all available in vitro and in vivo data and juxtapose this with clinical tissue exposures. So while this is beginning to have a positive impact in some companies, the overall picture across the industry is unclear. And in any event, there is always, firstly, room for improvement, and secondly, no room for complacency. But there does appear to be some complacency, and this was from an online survey run live during the last SPS meeting last year. And evidently 42% of participants considered that currently used preclinical assays and models are adequate to reduce attrition. I'd probably have some argument with that. But if we look at the current preclinical to clinical safety paradigm, this is flowing from the top of the slide to the bottom of the slide, starting at the stage where you've got project compounds. So this would be early lead optimization phase. You've already done some assessment on the primary target, but now you've got compounds, including tool compounds that interact with that primary target. And you can use conventional 2D in vitro safety assays, and also then more sophisticated 3D assays, microphysiological systems, and microelectrode array type platforms. And the readouts from this inform chemistry. Also, you can look at secondary pharmacology profiling of chemical series, and again, that informs the chemistry. And you could evaluate the secondary pharmacology risk using, again, these 2D and more sophisticated in vitro assays, and then move on to the preclinical in vivo phase, where you'll need animal ADME or DMPK data, and then on to human in phase 1 clinical trials. So what we've got in mind is to turbocharge the current paradigm. So we're not trying to, at this time, replace any in vitro or in vivo assays, but basically improve the knowledge that comes out of them, the data that comes out of them, the interpretation of it. So I've got some examples here, applying the SIVA toolkit to in vitro assays, applying secondary intelligence software to secondary pharmacology profiling, which I'll focus on, using SimCip Animal for the in vivo data, and then our our human platforms, the cardiac safety simulator, and of course SimCip. So our first area to address is secondary pharmacology, and here I'm going to hand back to Suzanne for an audience poll, have some participation.
All right, just one second, I will pull those up. All right, that should be launched.
Yeah, I can see that.
Okay, perfect.
So the first question is, for your secondary pharmacology screening, what data do you generate for regulatory submission? So you've got a selection of, you just have % inhibition of binding. You generate an IC50 or a Ki value. As well as an IC50 or Ki, you also do functional assays to find out for GPCRs if it's an agonist or an antagonist. You may skip that entirely and just run functional assays only, not do binding, or not applicable. So if you're dialing in and you don't work for a pharmaceutical company, please tick not applicable. So I think you can vote now.
Great, I see the responses coming in. We'll just give folks a few more seconds to vote, and then we'll close the poll and share the responses.
Okay.
All right, great. I'm going to close the poll and share the results.
It's interesting. Okay.
So it looks like most folks are doing IC50 or KI and functional assays.
Yeah. Yeah, I'm glad to hear that. I think that's a good way of doing it, but it shows also there's a variety of approaches, and it often is what's been traditional in the company or how it sort of fits in with the projects. So the next question is, do you use secondary pharmacology readouts only because regulatory agencies expect us to generate the data? Do you use them prospectively, in other words, to try and predict outcomes in vivo and in the clinic? Do you use them retrospectively? In other words, you're trying to interpret the readouts you're getting from in vivo studies in particular, and or not applicable. So try and think about if you use them both prospectively and retrospectively, try and think of the majority of the time that you would use them. So you can start voting now.
Great. We'll just give folks a few more seconds to put their votes in. All right, looks like most people have voted, so we'll close the poll and we'll share the results.
That's interesting. Yeah. So I'm glad to hear that as well, that the majority are trying to use the data to predict outcomes in the subsequent in vivo studies and in the clinic. So that's really useful to know. And also at the stage of regulatory submission, because I know that companies often sort of build up the panels throughout the discovery phase, but at the stage of regulatory submission, how large is your secondary pharmacology screening panel? Do you stick to the Bose 44 panel? Do you have more than that, between about 50 and 75 targets? Do you have 75 to 100 targets, more than 100 targets, or you don't work in a small molecule pharma?
All right, answers are coming in. It's great to see so many folks participating. All right, looks like most people have voted. So we'll share this last poll.
Oh, that's very interesting. So it's a bit of a split there between between all four options really, but between 50 and 75 and more than 100. Yeah, that's quite interesting. So again, it does vary between companies, and yeah, so right. So most pharma companies working on small molecule projects screen their compounds of interest against a broad panel of off-target receptors, and the annual spend on this on average is anything from about $500,000 to $1 million. So our question would be, is best use made of the data that you get back? So as you saw in the poll, the readouts can be used both prospectively and retrospectively. Regulatory agencies ask questions on the profiling data, and there's been a growing prominence of this discipline over the last decade. So here are some nice review articles. Some of you dialed in are authors on these, providing the pharma industry perspective. And there's also been a couple of nice review articles by the FDA authors in recent years. So we're focusing on the Bose panel. This was one of the reviews in the previous slide, and it's a consensus panel between 4 companies: AstraZeneca, GlaxoSmithKline, Novartis, and Pfizer, and it comprises 44 receptors that the authors agreed on in the panel. And that's 24 GPCRs, 8 ion channels, 6 enzymes, 3 transporters, 2 nuclear hormone receptors, and 1 kinase. And these were selected based on hit rate and the safety implications of hitting them. So even if you've got a larger panel, these are considered to be ones that you're likely to hit more often than not, and ones with the strongest association with safety implications. So it's an obvious panel for us to focus on. So for each receptor in the Bose panel, we have collated and curated the intended pharmacodynamic effects and side effects of reference drugs. So these are marketed drugs targeting the receptor for their therapeutic efficacy, as well as candidate drugs that are still in clinical development or were discontinued. But essentially, they've got both receptor binding data and also clinical data. And also for the principal pharmacodynamic effects, so by that I mean a reference drug that's targeting a disease indication. The principal PD effect is what you want it to do, but for your compound, if you hit that receptor, that becomes a side effect. But for these principal effects, we've constructed mechanistic safety pathways, and these are based on the format of adverse outcome pathways that are used in the agrochemical toxicology world. And the value of these is that they ensure the use of a standardized approach for each PD or side effect and each receptor or receptor interaction. Also, where available for each reference drug, we've sourced and expertly reviewed data on Ki or IC50, the lowest effective plasma concentrations, and plasma protein binding data. So there are 5 key aspects to the software. The first is qualitative information. The next is mechanistic insight, quantitative analysis, data visualization, and report generation. So on the qualitative information side, we've listed all the drugs targeting the receptor, including marketed drugs, drugs in clinical development or stopped or authorization declined, veterinary drugs, and research tools. We've also got a list of the main organs or organ systems affected, the commonest associated side effects, the liability of most concern, expected safety pharmacology outcomes, and translatable biomarkers. In terms of mechanistic insight, as I mentioned a minute ago, we've got mechanistic safety pathways for the most noteworthy side effects associated with the receptor interaction, each citing a useful paper or review article on the mechanism. For quantitative analysis, which I'll talk through in a minute, this is really a comparison of where your test compound sits in relation to drugs targeting the receptor for clinical efficacy. And then we have a range of data visualization tools, multiple ways of visualizing your data, including multi-compound comparisons. And then finally, report generation. So the software will auto-generate a report in Word, which you can edit in terms of tweaks to the conclusion or whatever. But all the hard work is done for you. So knowing the potential side effects from off-target activity of your compound is not enough. What you really want to know is how hard do I have to hit this receptor to see these side effects, or look at it from the other direction. How much engagement with this target can I get away with before I see this side effects? So are you down here or are you up there? This is going to vary between receptors and between pharmacodynamic effects of a drug at the same receptor in different tissues. This requires knowledge of how much target engagement is required by the marketed drugs targeting this receptor by evaluating the ratio of their effective plasma concentration, unbound, divided by the Ki or IC50 at the receptor. Then compare the predicted plasma Cmax unbound over Ki for your compound, where this is then an off-target hit. Does it sit within the range of marketed drugs, or do you have a margin? So this is kind of the table you would get from a company that you send your compounds to run a set of screening assays for off-target or secondary pharmacology. And the tabulation is in descending order of potency at the target. So at the top of the list here is alpha-2A adrenoceptor, where there's a Ki being generated with a Ki of 0.78 micromolar, and then the other hits are in descending order of potency. And what you can do is plot something like this, where you've got the log of the concentration, which can be the plasma concentration indicated by this vertical line, free concentration predicted in this case for your compound because you're not in the clinic yet. And then this table stacked up in descending order of potency as you move to the right. That's what we've got now. It's quite a good way of visualizing the table. But what if we could do this, where we could actually rate these interactions according to low, medium, or high likelihood of target engagement in clinical use? Would all the green ones be out to the right and all the red ones out to the left, where they're closer to the predicted free Cmax, or are they a bit of a mixed bag in the middle? So it's difficult to say without doing the analysis. This is what we want to achieve with secondary intelligence. So how did we build it? So for each receptor, so this is fictitious receptor X, and we had 6 drugs that we found that were had clinical data as well as Ki data, and they all have a set of side effects. So using FAIRS analysis and also other reliable sources of side effects, what we've done is to look for side effects that all the drugs have in common, because then you can be fairly sure that it's an on-target effect at that receptor and not an off-target effect at another receptor by some drugs. And so we've got in this panel the receptor X, and we've got 2 sides to the table. One is a positive interaction, so for GPCR an agonist effect, and then a negative interaction, an antagonist or inverse agonist type effect. We've listed the main organ systems affected, the liability of most concern, the commonest side effects, expected safety pharmacology outcomes, and the translatable biomarker. And we can also, for each of these 3 adverse effects, construct a mechanistic safety pathway. So these start with the receptor interaction on the left and then step up in complexity. So it goes from receptor interaction to cellular response to organ level response, and then whole organism response. In this case, it's explaining how an antagonist action of this receptor leads to headache, and then there's a reference with the PubMed ID at the bottom that you can actually back up this pathway. Same for dizziness, same for hypotension. So in terms of building the plots for each drug, we've obviously got the molecular weight, and we need to find the plasma protein binding, and we then have to find the lowest effective plasma concentration in human, taking care to identify that we've got an effect that's the lowest concentration causing a measurable or statistically significant effect, and not a concentration below there's basically a no effect level, or one that's high up the concentration response curve. So it's really around the threshold level. We've got the Ki at the receptor, or IC50 for some targets. So the PK/PD data predominantly from healthy volunteer studies, and the Ki or IC50 data predominantly from IUFAR BPS source references. And what we've got in the equation is the effective plasma concentration unbound over the Ki or IC50, and then we can plot these as we've got for the other drugs as well. So a total of 6 drugs here. This is the plot for drug A. And what we're interested in really is the left-hand drug in the plot, and I'll just talk you through that here. So this plot is probably the other way round from what we're used to in safety pharmacology, looking at margins. So think of it as an inverse margin. This is about coverage. So here the effective plasma concentration unbound is equal to the Ki. So there's a ratio of 1. If you move in this direction, your plasma concentration to get a measurable PD effect from the reference drug is greater than its Ki. And if you go further out, it's far greater than the Ki. There are certain reasons that could explain that. If you go in the opposite direction, here to achieve a pharmacodynamic effect, your reference drug plasma concentration or free plasma concentration doesn't need to be the Ki. It's lower than the Ki value. And out here, it can be much lower. And this is where we get suspicious if we see this. So are we looking at a local delivery route, for example, inhalation? So you have to make sure you're if you're doing that, the plasma concentration has to relate to a response that is accessible via the circulation, and not so for example, if it's inhalation, you might be looking at effects on heart rate with, say, a beta-2 agonist, not an effect on lung function. It also might be explained by pharmacologically active metabolites, or PAMs, which I will walk you through. And we've been dealing with these. We hit quite a few of them during the course of this project, and I'm going to show you an example for lefepramine, which is a norepinephrine transporter inhibitor. So here we have the plot. This first point here is the coverage for the parent drug. So this is data from plasma concentration of the parent drug, unbound, divided by its Ki. If you were to ignore the existence of pharmacologically active metabolites for this reference drug, this is where you would plot it. However, when you take them into account, this is the actual metabolite on its own, and this is the combination of the two. So this is an aggregate concentration of the two activities divided by their respective KIs. So if you look at the red dot, circle dot, and the purple circle dot, which is the data we would use, there's a 2 log order shift to the right. So if we stuck with the original, just use the parent concentration, we'd be getting quite erroneous data. But to enable this, we need to source the Ki, plasma protein binding, and simultaneous plasma concentration of the pharmacologically active metabolite. And if any of this information is missing, we can't use this reference drug. So we can't just say, oh, we ignore the metabolism and the pharmacologically active metabolite. We have to drop this from the list. And also to point out that the eventual position of the combination data point depends on the relative potency of the parent and the PAM, the plasma concentrations of the parent and the PAM, and the plasma protein binding, which differs between the parent and its active metabolite. And another interesting thing to point out is the impact on slow and fast metabolizers will vary between drugs according to the above parameters. If the active metabolite is predominating, this is going to have a different effect than if the parent is. And we've encountered and dealt with over 25 reference drugs so far with pharmacologically active metabolites. So it's certainly at least double the work on over reference drugs that don't have this issue, but it's something that you have to deal with. I'm also going to show you this. Now, this shows the comparison of ranges of unbound plasma concentration of Ki over Ki across different receptor classes. And in the notes, I've said this is a relative positional plot, and I've deliberately removed the x-axis scale because I know you'll all be snipping away. And I don't want to give you the answers that we've got in secondary intelligence to help you say, oh, thanks, Will, we'll settle for that. But it is interesting that there are differences between the classes, but it's also clear there are no obvious rules of thumb. And within each class, different receptors sit in different places. So you really need to do this receptor by receptor. So just to walk you through how it operates. So we'd have a plot of reference drugs, and the red zone, which is the high likelihood, starts at the left-hand drug and goes upwards. And then we've got, you can set this amber region to a half log or a log, depending on how you feel about the data and your own risk appetite, if you like. And then you have the low risk area. So if your compound ends up in here, this is what I would describe as the cat amongst the pigeons. Your compound is in amongst the reference drugs, so that there is a high likelihood that you're going to see the pharmacodynamic effects and side effects of the reference drug, which of course for your drug are all side effects. If it ends up in the medium zone, you're clear of the left-hand drugs, so it's got a medium likelihood. We've classed it as that to allow a bit of uncertainty, but also if your predicted free Cmax, because it's all based on predictions at this stage, if that creeps upwards, you may end up in the red zone. And then if your compound is happily in the green zone, then it's probably not going to require you losing any sleep over it going forwards. So we have a receptor panel view. So this is the Bose panel laid out a bit like the periodic table. And when you've run your readout through it, effectively the receptors that have been hit light up, and they indicate with this symbol with a plus sign indicates it's got an agonist or a positive interaction. The cross sign, diagonal cross, indicates a negative or antagonist effect. And we also have not defined, where you don't know what the functional outcome is yet. And then we've got green, yellow, and red, also with letters to try to help if you've got red-green color blindness with high, medium, and low interaction. And then you can click on the receptor, which will take you to an individual panel view. So here we've got a list of the effects, as I showed you earlier, and also a list of drugs targeting the receptor, which you can scroll down, and it's got links to Kemble and the IUPHAR, BPS database, etc. In this panel on the top right, you can alter the unbound plasma concentration prediction to see what will happen if it creeps up, or you could perhaps put in what concentration you might be expected to see in phase 1, which will be higher than your clinically expected plasma concentration. And in the bottom here, you've got a plot. So here the red zone starts at the position of the left-hand drug in the cluster of reference drugs. This is the where the ratio is 1. And your drug has unfortunately ended up in the red zone, so it's likely to have this activity in clinical use. We also have different visualization types. So here we've got the interactions in descending order of likelihood. There was the plot that I showed earlier with the scale. The X scale is just concentration in micromolar, and that's the predicted free Cmax for your compound. We can, or you can also compare different compounds up to double figures, and also switch the arrangement of the receptors. And this plot here, this is a single compound, but it shows you how far into each zone you are for the interactions with off-target receptors. So in terms of interpretation, so this actually uses 3 different receptors in this, just to give you a bit of variety. But here, the test compound is into the green zone, which is probably started around here. You can just about see it where my pointer is. The likelihood of interaction with this off-target receptor in clinical use is low, and so we can only suggest next steps, but I would probably suggest that further investigation was probably unnecessary. Here in the amber zone, you've got a medium likelihood, so the red zone starts just about here, where that left-hand drug is plotted, and you've got a 1 log width medium zone, and your compounds ended up there. So what I would certainly suggest is that you investigate further if the predicted free Cmax increases, but you might decide you want to investigate anyway, just to be on the safe side. Then finally, so you can see here the test compound is in amongst the reference drugs. So you might want to consider further investigation of the potential safety issue in vitro and or in vivo, and this could involve additional endpoints that you would incorporate into safety pharmacology and or toxicology studies, or set up bespoke studies to follow it up. And the software will also auto-generate a report if required, which could be purely for internal use, or you might even want to append it to a report of a screening panel and form part of regulatory submission. And just to give you the flavor of secondary intelligence in numbers, so it addresses all 44 receptors in the Bose panel, which is with the GPCRs having 2 directions of interaction, is around 70 in receptor interactions. There are over 700 reference drugs which we evaluated in our initial evaluation, from which 150, or rather over 150 reference drugs have been plotted, and over 100 mechanistic safety pathways have been prepared. I did a quick wordle on the commonest associated side effects. Pulsar de pointe is nowhere to be seen, but the commonest, as you probably know anyway, are nausea, headache, and also hypotension. So you know, tachycardia might well be reflex. So these are commonly seen. Dyspnea is another common one as well. So and that's been quite useful in itself. Often adverse effects that are often to some extent ignored or given a lower status. So I also thought it was a bit of fun to just talk you through a secondary pharmacologist's life before and after secondary intelligence. This is before, so a senior manager might ask or say that in this fictitious project called GHA, I see our favorite GHA compound as some antagonist activity at 5-HT3 receptors. Yes. What are the safety implications? I'll need to check our knowledge document and get back to you on that. What are the marketed drugs targeting 5-HT3? I'll just have a look at Wikipedia. But will it even reach plasma concentrations in the clinic that will interact with 5-HT3? That's difficult to say. We'll have to compare it to the marketed drugs, and this will take a while to do. So that's before. This is after. Same questions. So I see our favorite GHA compound has some antagonist activity at 5-HT3. Yes, but actually, what are the safety implications? Constipation is the main concern, but actually, what are the marketed drugs targeting 5-HT3? Allosetron, azositron, solancitron, dolasetron, all the cetrons. But will it even reach plasma concentrations in the clinic that will interact with 5-HT3? It's actually well into the green, so we shouldn't have to worry about that. Why didn't you say that in the first place? So in summary, secondary intelligence addresses off-target hits at the 44 receptors in the Bose panel. The software is available now, and there are 3 ways of accessing it. Firstly, you can join our consortium, and additional receptors up to a final total of 200 will be included, plus other in silico tools that are ready now. Also, we hope to develop further QSTS tools guided by consortium member needs. The second way of accessing it would be to purchase an annual software license. And then the third way is via consultancy projects. So we would run your compound or compounds through the tool and provide a report. And you can contact me for further information. I would like to thank colleagues within the team, within QSTS, within the software team, and our excellent library services. Senior managers have been very supportive. We've worked with 2 external consultants, Chris Pollard and Lynn Rosenbrier-Ribeiro, who are former AZ colleagues of mine. We've had help from Michael Spedding and Elena Facenda at IUFAR to clarify where they get their data from. Other Certara colleagues, and of course colleagues at Amgen who've provided really helpful feedback on the tool. So thank you for your attention and for time dialing in, and I'm happy to field questions that you might have.
Great, thank you so much, Will. We'd like to invite our audience to ask your questions to Will. You can just type those in the chat, or sorry, in the questions panel, and we'll try and answer as many as we can in the time we have. So Will, someone wants to know, what data do I need on my compound to enter into the secondary pharmacology software?
Ideally, you need to have a knowledge of the predicted free plasma concentration, and from the secondary pharmacology readout, a Ki value, or if the assay only generates IC50 and IC50 value. I know that some companies, perhaps at an early stage, just generate % inhibition. If you've got a % inhibition of at least 10%, but probably more safely at least 20%, then you can estimate the Ki from that data, and we can certainly help with a little formula to do that, and then you could put those data in. But our data works off Ki, not % inhibition.
To use the software, do I need to know if my compound is an agonist or an antagonist at a particular receptor?
Well, obviously that is important to know, but if you don't know that yet, we have the option of where the interaction is not known, as you saw in the symbols, the open circle. But what you can also do is enter your data in two ways, both as an agonist and an antagonist. What you'll then have is the possibility that, say, an agonist interaction might give you a red readout, and an antagonist might be green, and then it becomes obviously important to know by doing a functional assay which interaction or which direction it is. Whereas if they both came out green, then to some extent you don't need to know anymore. If they both came out red, you might want to still find out which direction it is, because obviously that affects the side effect profile. So it would actually help you decide if you have that strategy of doing just generating KI first and then doing functional. You could actually use it to help you decide which assays you needed to repeat on that compound to get a functional assay, functional readout.
Great. Someone wants to know, what data set did Certara use to train and develop secondary intelligence? Can we check the chemical space that is covered by the data set?
Yeah, I think train is the wrong word, because it's, we're not, if you like, not using machine learning. To build the data set, we've used reference drugs that are selected for the receptor and that are used clinically. So it does cover a reasonable amount of chemical space across 44 receptors. But we can provide you with a list of reference drugs if you wanted to check that. But that's an interesting question. So if there's over 150 plotted, it's whatever the chemical space it occupies, which is clearly a finite amount.
Okay, great. I think that this appears to be all of our questions. So thank you so much, Will, and thank you so much to our audience for those excellent questions and feedback. Will, if you want to go to the next slide, please. Just have a few announcements. On behalf of Certara, I'd like to thank everybody for attending today's presentation. This concludes the webinar. Goodbye and have a great day.
Thanks, everyone. Bye.